Welcome to our channel. For decades, the science behind those tiny crystal-like glands on cannabis, well, it seemed pretty straightforward. But some groundbreaking new research has just revealed a biological process so bizarre, scientists are calling it alien.
So let's just dive right in. The central mystery here is the trichome itself, that tiny glistening little structure on the surface of the plant. How in the world can something so small produce such an enormous volume of complex chemicals with almost perfect efficiency? I mean, this question has stumped scientists for years, mainly because the answer we all thought we had, it just didn't add up.
For the longest time, the accepted science was, elegantly simple. The trichome was just seen as a balloon on a stalk, basically a simple storage head sitting on top of a pillar.
The theory went something like this. Inside that trichome head, you had a little disc of about 8 to 16 secretory cells that acted like tiny separate factories. Each one was thought to be working all by itself, slowly releasing its product into a shared space. It sounds logical, right? But the evidence just wasn't backing it up.
Yeah, the math just wasn't mathing. The balloon on a stalk theory was full of these biological plot holes that science simply could not explain. It was like the factory was running way too perfectly for the blueprints we were looking at.
So this old model created three huge puzzles. First up was speed. Production was happening way too fast for a bunch of separate cells. Second, the coordination was flawless, as if every single tiny factory was in constant communication. And third, the output wasn't steady. It would spike dramatically at the same time every single day. It just made no sense for a collection of disorganized, individual cells. And this slide really shows you the scientific dead end we were at. On the left, you've got the old model, a bunch of slow, walled-off, inefficient little workers. But on the right, that's the reality we could see but couldn't explain, a hyper-efficient, perfectly synchronized factory. There was a giant piece of the puzzle missing. So here's the turning point. Breakthrough research, conducted between 2022 and 2025, using brand new microscopy techniques, proved definitively that the old model was just wrong. The balloon on a stalk theory is officially dead. Please like and subscribe, it really helps our channel a lot. Alright, so now let's get to the new reality. What researchers discovered was something, well, far more bizarre and way more sophisticated. It wasn't a collection of cells at all. It was one single, massive, coordinated entity, a supercell. This right here is the key. The proper scientific term is a syncytium. What happens is, as the trichome matures, those individual secretory cells actually dissolve their own internal walls. They fuse their insides together. They become one giant unified cell with multiple nuclei, a true biological megastructure. And this crazy architecture solves all the old puzzles. These cytoplasmic bridges create a superhighway for materials to move at unbelievable speeds. The whole thing transforms into a single, massive factory floor. And crucially, this factory is polarized. All the internal machinery, all the little organelles, they all migrate toward the top, creating this incredibly efficient assembly line that's aimed directly at the storage space. But this factory doesn't just run on a whim. It develops and it matures on a very strict schedule, a predictable life cycle that completely dictates its productive power. Scientists have actually mapped out three key phases. You've got phase T3, which is basically the construction phase, where the plant is just building the supercell's infrastructure. Then you hit T6, the secretory zenith. This is the absolute peak of production. And finally, there's T8, the late stage, where production slows down and the focus shifts to just maintaining what's already been created. And during that T6 phase, the supercell is an absolute powerhouse. The trichome head swells up to its maximum size, averaging about 72.2 micrometers, or around 0.003 of an inch. It's visibly swollen with resin as that factory is just running at full capacity.
Now here's where it gets even more fascinating. The supercell doesn't just run 24/7. Its entire operation is governed by a strict circadian rhythm, tied directly to the daily cycle of light and dark.
See, trichomes can't make their own food. They're what scientists call a sink. They are completely dependent on sugar fuel that gets sent up from the leaves. So as the plant soaks up light all day, it creates this sugar surge that peaks late in the day. And that's what kicks the supercell factory into overdrive. The incredible insight here is that the bottleneck isn't the machinery. A lot of the time it's just sitting idle. It's the fuel supply. So understanding this complex rhythmic biology, it's not just an academic exercise. It completely rewrites the playbook for anyone trying to cultivate the plant. It really transforms growing from an art into a science of managing these microscopic factories.
And a huge part of this is realizing the stalk isn't just a pillar holding things up, it's the supercell's umbilical cord. It's a regulatory hub that can sense the plant's overall health, it manages hormone signals from things like drought stress, and most importantly, it controls the flow of that precious sugar fuel into the factory head.
This new science gives us some really clear, actionable strategies. First, timing your harvest for the late afternoon means you can capture the peak of that daily production cycle. Second, you can use specific light spectrums, like far red light at the very end of the day, to basically signal the supercell to pull in even more resources. And third, applying strategic, controlled stress, like a brief drought, can trigger hormonal signals that ramp up production of protective compounds.
And the stakes for getting this right are enormous. We are not talking about tiny little percentage gains here. Research shows that the levels of those volatile terpenes, the compounds responsible for the aroma, they can fluctuate by an incredible 1,500% based on the time of day and the temperature in the room. So moving beyond that simple balloon on a stalk model isn't just a small correction, it is a complete paradigm shift that opens up a whole new frontier in understanding this plant.
And there's one last alien revelation for you. The final, critical step, creating the main cannabinoid precursor, THCA, doesn't even happen inside the supercell. The factory builds all the parts, and then it exports the final assembly enzyme to finish the job outside its own walls. It's a brilliant way to protect the cell's machinery from its own powerful products.
This new understanding also explains the huge increase in potency we've seen over the last few decades. Without even realizing it, breeders have been selecting for plants with bigger, more robust supercells. Landrace strains often had these smaller stalks supporting maybe 8 fused cells. But modern cultivars, they now have much larger stalks that can support supercells of 15 or more, with a wider umbilical cord to transport way more fuel.
As the source material puts it so clearly, the era of the balloon on a stalk is over. We're now seeing the trichome for what it truly is, an absolute masterpiece of biological engineering.
And it really leaves us with a fascinating final thought. This entire biological model has been completely rewritten in just the last 3 years. If our understanding has come this far, this fast, what other alien biology are we about to uncover next? Thanks for watching. This content does not constitute medical advice, legal advice, or recommendations for the use, self-treatment, or consumption of any substance.